Today we are welcoming Dr. Gretchen Hansen. Gretchen is an assistant professor in the Fisheries, Wildlife, and Conservation Biology Department at the University of Minnesota, and she is also a researcher here at MASERC. Today she will be sharing her recent MASERC project testing a citizen science approach for testing aquatic invasive species using eDNA.
Great, thanks. Thanks for being here, everybody. I'm excited to talk to you about the results of our study that we carried out last year, examining how well volunteers, a lot of you who are here, could carry out eDNA sampling to detect aquatic invasive species in our Minnesota lakes. So I just want to start by thanking MASERC for the funding and for putting on this webinar, and also thanking my co-authors who are all listed here, who helped with the development and execution of this project. And of course, thanking all of the volunteers, we'll come back to that at the end, who dedicated their time to actually carrying this out. So this is obviously couldn't have been done without you. Okay, let's make sure we can go to next slide. There we go. Okay, so probably don't need to say this to this crowd in a MASERC webinar, but sort of the impetus for this project is the idea that aquatic invasive species are really widespread across the landscape of lakes in Minnesota. Many different kinds of aquatic invasive species. The map I'm showing here just shows 3 of them. But they're really widespread in lakes throughout the state. Hundreds of lakes in Minnesota are known to be invaded by 1 or more aquatic invasive species. And of course, we know these species can have impacts, ecological and economic impacts on aquatic systems. And knowing where they are, their distribution in lakes and rivers throughout the state can help to enable early detection and possible control and containment efforts, and can also help us assess the effectiveness of existing management strategies if we can kind of track the spread. But we don't really have a reliable system of tracking where these species are in an unbiased way. So I would argue that we don't really know how widespread aquatic invasive species are in Minnesota lakes, just because of the way that they are documented and sampled. For the most part, this isn't 100% true, but for the most part, aquatic invasive species are sampled opportunistically in Minnesota lakes. So that means when somebody observes an invasive species, either a person who's out on the lake who reports it to the DNR, or maybe a DNR or PCA or county level sampling event. When it's observed, then we document that the species is there. But we're not necessarily going out in a strategic way to the majority of our lakes to test whether these species are there. So the sample is not random. It's biased towards highly visible species and lakes that are visited really frequently, a relatively small subset of lakes relative to the number of lakes that are in Minnesota, and likely is an underestimate of the true prevalence of aquatic invasive species in lakes throughout our state. So why do I say that? There was a study in Wisconsin, I guess 9 years ago, that examined what do you find when you sample randomly. So sample in a way that is statistically valid to tell you about the whole population of lakes in Wisconsin, in this case, not just the subset of lakes that species have been reported. So they designed a probabilistic survey to go to different kinds of lakes, stratified by whether they were big or small or had boat access and a couple other things. What they found was when they went out and surveyed in that way, they found AIS prevalence about 5 times higher than what they had previously documented using a system that's similar to what we do in Minnesota with kind of opportunistic reports. So by going out and actually looking in a strategic way, you find prevalence that is quite a bit higher. And again, this can kind of bias our understanding of where species are, potentially reduce our management effectiveness, and bias our risk assessments of knowing what kinds of lakes support what kinds of species. So this kind of probabilistic random survey method could tell us a lot about where species are in Minnesota lakes. But of course, there are a lot of them. Minnesota has thousands and thousands of water bodies throughout the state. And it is very difficult to go out and sample each of those, especially using what I would call more like traditional or standard methods of detection. So that Wisconsin study, they used physical gears to look for invasive species. So they were setting traps, they were doing scuba and snorkel surveys, all kinds of transects on the lake. And that just takes a lot of staff time, a lot of travel. It can be really expensive to sample lakes in that way, especially when you're looking for something that is likely to be pretty rare, like an invasive species that has not yet been documented. There's likely to not be very many of them. So it's pretty labor intensive to go out and look and tell yourself, like, is this lake truly uninvaded? How much sampling do we have to do to tell us that a species is not there, or to make it likely that we would find it even if they're there in small numbers? So this led to my interest in environmental DNA, or eDNA, as a potential method for cost-effective widespread monitoring for AIS. So eDNA, I'll say that a lot, so that's environmental DNA, is just DNA that's released by an organism into its environment. So in this case, DNA that is released by an organism into the water of the lake in which it lives. The use of eDNA is increasing kind of worldwide for all kinds of monitoring approaches, not just AIS, but AIS is one potential use. eDNA can be a really sensitive method, so you can detect species at really low numbers if you can get and filter their DNA from the water. You can use it to detect multiple species at potentially low densities at a relatively low cost relative to going out and doing extensive physical surveys across a lot of lakes. And so a few years ago, my co-authors and I started on what I'll refer to as phase one of this project. So our first approach was to really evaluate a couple different things about eDNA in terms of how effective it is at detecting invasive species and what factors might influence the effectiveness. So in phase one, we had a couple different objectives. One of them was to look at seasonality of detection of the 4 target species that we were interested in. In this study, it was spiny water flea, common carp, zebra mussels, and rusty crayfish. And we picked those for a few reasons. One of them was because of this seasonality question. We wanted species with pretty different life histories, so different ways in which they inhabit the lake and seasonal patterns in which we would expect their DNA to be more or less prevalent. And so we picked these 4 species, and they also had good DNA assays already available. So we knew we could use eDNA relatively reliably to detect them. So we wanted to say, does it matter what time of year you go out and sample in terms of how well you can detect these 4 target species? That was one of the questions for our phase one. The quick answer, this webinar isn't about that phase. I'll just say that the short answer to that question is yes, it does matter what time of year you go out. Most of these species exhibited a relatively high level of seasonality in our capacity to detect them using eDNA. So this figure here shows the seasonal curves of each of the species. We don't need to go into details, but you can see this purple line is zebra mussels. And they were the most detectable species in phase one. We were most able to detect zebra mussels, and we were best able to detect them in kind of midsummer. And a couple of other species followed a similar pattern. It wasn't exactly the same, but somewhat similar. So that's important when we get to our protocol for the volunteer sampling. We also tested a number of different field and lab methods to understand how those different methodological choices influence the detectability of the species. I'm not going to go into great detail about those. A lot of those are related to lab methods, but also some field methods like surface water versus deep water, near boat launch versus away, et cetera, et cetera. So we did learn that a couple things about what kind of filter to use, how much water we want to volume, how much water we need to sample to maximize our probability of detection, and what locations within a lake were important. And that last one was important for this volunteer study because we learned at locations kind of throughout the lake, whether it was deep or shallow, we were pretty equally able to observe these species. And so that was important in our design of this volunteer model. So not going to go into all the lab methods, but we did test a bunch of things to figure out how can we optimize detection. Okay, so we learned a lot from phase one about when and where and how to sample to maximize our detectability of these target species. So then we wanted to say, okay, taking that knowledge, how could we start thinking about sampling more representatively lakes throughout the state? And that led us to be interested in whether we could potentially tap into the network of interested Minnesotans who might be interested in helping detect invasive species in lakes that they care about. And the AIS Detectors program run through MASERC and Extension here was a model of tapping into the vast number of people throughout the state who are interested in doing this stuff. And we wanted to examine whether we could use volunteers to collect eDNA samples. And one of our objectives was to think about if we were going to scale this up to a broad scale, how could we design a program that might potentially be scalable in that way? So our objectives for this project were to first develop training materials and protocols for implementing volunteer monitoring using eDNA, and then kind of test that protocol out, put it out in the field to you all, to the volunteers, and see how accurate eDNA monitoring could be when conducted by community volunteers without somebody on site to do the training. So the first part of our goal, we spent the first part of last winter and spring testing out different kinds of methods for collecting water and pumping water and developing training materials that did not require on-site supervision or training. So we wanted to develop materials that could be accessed kind of anywhere by volunteers. And so we came up with a written sampling manual that did go out to all the volunteers as a paper copy. It's also available on our MASERC website. And we developed a series of videos showing the different steps of how to sample. And those are all available on the YouTube playlist from MASERC. So we developed these training protocols and then kind of put them out there with instructions to the volunteers. We also put together volunteer sampling kits. As I said, we tested different kinds of pumps and components of this kit in the lab and in the field here at University of Minnesota Twin Cities before deciding on exactly what kind of equipment we wanted to send out. And then we sent all this out with data sheets and the manual and all these little components in the kit. So what did volunteers do after they signed up? After they were assigned to a lake, they received their kit. They were instructed to review the training material, both the videos and the written instructions, and then to sample the lakes that they were assigned to, that they had signed up for, during a specified collection window. So during a specified time, which was June 30th to July 9th. So we gave them all the materials, we gave them the instruction, we gave them a map and a window of time to go do it. We also assigned a location. So we did kind of random assignment of locations based on where public access to the lake was. But we also told volunteers that if they had their own access, like via a dock that they could sample off of, or if they were just out on the lake in their boat, they could feel free to sample at a different location. We just wanted them to tell us where that was. So this map shows an example where we assigned the different volunteers locations based on safe public access. So mostly boat launches, but sometimes there were some parks with fishing piers and stuff. But again, gave the flexibility to say, if you want to sample somewhere else, that's fine. You just need to tell us either by tell us what the coordinates was, or even just draw it on the map of where you went. Each volunteer or each lake was assigned 10 samples. So in theory, that could be 10 different volunteers who went out to 10 different points in the lake. In reality, a number of our volunteers signed up for more than one sample. And, so we had sometimes one volunteer taking multiple samples within a lake, but we got 10 samples per lake from each of our included lakes in the study, which are listed here. So what they did was collect water and filter it from their site, and then following the instructions, and then send back to us in the lab the filter that they had run their water through. And then we, the researchers back at the universities, analyze those filters for the presence of the DNA of our target species. We also asked volunteers to ship their kits back to us. So we provided shipping and boxes for that so that we could potentially reuse some of these materials for future sampling. And then we also asked volunteers to complete a post-sampling survey. Again, we had a paper copy or an online version of it. And this survey was focused on kind of how easy it was to sample, how well people understood the instructions or any difficulties they encountered, ways in which we could improve our training and our instructions for any future endeavors. Okay, so what did we learn? Number one, incredible response rate. So I don't do social science research as a normal part of my job, but I do hear about a lot of it. And I know that response rates in the 20 to 30% are considered pretty good. We had 85 out of 100 survey responses sent back to us. I think that's super high. We had 98 out of our 100 filters returned. So 98 out of our 100 samples came back to the lab and were able to be used, and 91 out of 100 kits returned. So this is just an incredible response rate that really gives me hope about how we could potentially implement this in the future. And kudos to all of the volunteers for their really great follow through. One thing that we learned both from the surveys and from 91 out of the 100 kits being returned is that our instructions as they related to shipping things back was a little bit of a unclear to some volunteers. We did not make our instructions clear enough because I think this was because there were two things that volunteers were instructed to send back to us. One was take the filter from your filtered water, put it in a certain envelope, send that to the lab. Number two, send the box of materials and equipment back to us. And that got a little confusing sometimes. So that's something that we could do better at. Okay, so how about detection? How did the volunteer sampling do in terms of detecting invasive species in these lakes? So I'm going to preface this by saying this study was designed as kind of a proof of concept to assess the detectability of these invasive species using this kind of volunteer monitoring. So we used lakes where we knew already the status of invasive species. We were not going out to say, let's find new species right now with this sampling. We were asking questions about given that we know this invasive species is here, how frequently is it detected in the volunteer samples? So that's a key point that I'll just keep emphasizing. We're not looking for new detections with this. We are just assessing the probability of detection using this method of 10 volunteer samples spread throughout the lake, taken in summertime, most of them from near shore. Okay, so I'm going to show you a series of graphs and I will tell you what they mean. So we'll have a graph for each species. The first one here is common carp, which were only sampled in one lake in this phase of the study, Mille Lacs. So again, we know common carp exists in Mille Lacs, and we want to look at the probability of detection, which is just the proportion of positive hits on DNA that we got from the 10 volunteer samples. And then the dot, so that's what the bars on all of the figures I'm going to show you are the detection rate from volunteer samples. And then the dots are the detection rate from phase 1 in the samples that correspond to about the same time of year. So hopefully that makes sense. So here in Mille Lacs, we had a detection rate of about 10%. So 1 out of the 10 samples from Mille Lacs was a positive for common carp. And that detection rate was even lower in the phase 1 samples for that lake. So volunteer sampling had a higher detection rate for common carp in Mille Lacs than our phase 1 sampling did at approximately the same time of year. It'll get a little bit more interesting as we go to other species. So here we can see rusty crayfish, which we knew were present in 5 of our lakes. So again, the colored bars show how well they were detected in the volunteer samples across different lakes. And the black dots show how well they were detected in our phase 1 sampling. So here we can see a couple different things. The probability of detecting rusty crayfish was pretty different across each of the lakes where we know that they exist. And it was generally higher, probability of detection was generally higher using the volunteer samples than in the phase 1 sampling. So I want to put a caveat to this that it's not totally comparable. The phase 1 sampling, as I said, looked at different locations within the lake, deep versus shallow samples, lots of different times of year. The labs also got kind of some different protocols for improving their detectability by the time we got to the volunteer sampling. So it's not really a total apples to apples comparison, but I put it on here just to kind of emphasize how well the volunteer monitoring worked for detecting these known populations of species. So there was one place, Lake Owasso, where we had detected rusty crayfish at low numbers in phase 1, but they were not detected at all in the volunteer sampling. So that's a notable kind of exception to the high detectability across the other lakes for rusty crayfish. Next up is spiny water flea. Spiny water flea were by far the hardest to detect using eDNA in our phase 1 sampling. This has been shown in other studies as well. There's been some studies out of Wisconsin showing that eDNA detections of spiny water flea can be challenging. And again, we saw some kind of mixed results from the volunteer samples. We saw relatively higher detectability in Lake Vermilion and Mille Lacs, but from the volunteer samples, but we still had 2 lakes where we know that these species are present that we did not detect them in the eDNA samples. And that was true both with the volunteer samples and with our phase 1 sampling. So this sort of reinforces that spiny water flea are less reliably detected using eDNA than our other species that we looked at. But volunteer sampling was able to detect them in a decent proportion of cases in Vermilion and in Mille Lacs. And our last species is zebra mussel. In both the volunteer sampling and in our phase 1 sampling, zebra mussels were the most detectable species. They were most reliably detected in places where we know that they exist. That was true here for the volunteer sampling. Zebra mussels were the most highest detection rate. So in Forest Lake, we had 100% detection rate. Every single sample taken by volunteers did show positive for zebra mussel. No other lake was 100%, but they were all pretty high, like hovering around 75, 80%. And again, our probability of detection in phase 1 was relatively high for zebra mussel across these lakes as well. And again, one other exception where Lake Owasso, the volunteer samples did not detect zebra mussels, but the phase 1 sampling did. And in Lake Owasso, they're at really low numbers. I think maybe only villagers have been found, or possibly there's one adult or 2, but they're at really low numbers in Lake Owasso. We did detect them in phase 1, but not in phase 2, or not in the volunteer sampling. But across the board, pretty good detection rates for zebra mussels in both of the studies, including with the volunteer monitoring. So this just sums up probability of detection across all lakes. What is the average in phase 1? So our initial study is in this sort of orangey salmon color, and in the volunteer sampling is the turquoise color. So for all of these species, volunteer sampling on average detected these species at a higher rate. Again, I'll re-emphasize, it's not completely apples to apples for a number of different reasons related to both field and lab methods, but really encouraging to see these high detection rates from the volunteer samples. Okay, I'm going to touch on contamination. This is an issue in any eDNA study. So eDNA is valuable because it's so sensitive. It can detect DNA at really, really tiny amounts. So that makes it a powerful tool for detecting rare species like new invasions, for example, but also makes it prone to potential contamination events. So because just tiny, tiny amounts of DNA can show up in the results, if the sample at any phase from field to lab is exposed to alternate sources of DNA, by somebody touching something and then touching something else, or there's DNA on potentially the boat that you're using for sampling because it came from another lake, there can be contamination in the lab, et cetera. Contamination is always an issue when you're using eDNA. And there are a number of steps you can take to avoid contamination, wearing gloves when touching or using any of the equipment, being really careful not to touch any other surfaces while you're wearing the gloves, leaving the equipment in its sealed packaging until it's ready to use, leaving the sample bottle closed until it's ready to use. all of those are steps you can take in the field to reduce contamination. And we bleached all the equipment before sending it out to remove any existing DNA. All of these steps can be taken to reduce contamination, but it can still happen through a number of processes that we may or may not even know what they were. So one of the ways that we measure contamination is by testing what we call field blanks. So this is just water, like distilled water that has no DNA of any kind in it, that when we do our studies, we always bring a field blank out with us for every sampling event. So we bring a jar of water out, open it up in the field, and then close it, and then treat it the same as we would any other sample. And if you detect any DNA in your field blank, that is an indication that at some point in the process, you introduce some contamination. So we weren't able to have every volunteer for every sample do a field blank, but we did send 10 volunteers who were randomly selected a field blank in their kit, and instructed them to open it up in the field and then do an additional filtering and send it back to us, so that we could, it wasn't a total, we're not testing everybody's contamination rates, but it was a random sample of 10 people, one from each lake, to see what kind of contamination rates we got. And what, this graph is funny because there is only one point on it, but what we found was only one contamination event across all of our blanks. So we had 10 blanks and we tested them for most of the species. We didn't test for common carp in one half of the blanks, but when we did that, we found just one blank that had zebra mussel DNA in it. And so pretty low contamination rate there, and no other blanks had any contamination of any of our target species. So this is notable that it's a pretty low contamination rate, especially for volunteer sampling. But there is some contamination happening in the field, and that happened in our phase 1 of the study as well. So this isn't a dig on volunteer samples. It happens in every eDNA study. It's just important to recognize it can happen and can make you sort of interpret some of the results with a grain of salt. But overall contamination rate was quite low from the volunteer samples. Okay, and then our last objective that we wanted to do and get information from volunteers was get feedback from that survey on how the sampling went and any suggestions for improvement in the future. So what did we learn from the volunteer feedback? We did hear a lot of great reasons of why people wanted to volunteer in terms of their motivation for being involved. Those are listed here. We learned that most people found the training results and the process relatively easy to follow. Most volunteers felt like they did a good job, that they were easily able to find a sampling location, that collecting the water was pretty easy and pretty straightforward. We did recommend that everybody sampled with a partner for safety reasons, and we found that the majority of volunteers did not sample with a partner, and that will come up in a subsequent slide here. Most people said sampling equipment was easy to use, although that was a little bit lower at 88% than some of the other responses. So I think we could work to improve that a little bit. And 100% said they would participate in eDNA sampling again. So that was really great to see. We learned that about half of the volunteers went to their pre-assigned site. 20% went to a private access that they were able to get to. A little less than 10% went out in a boat to collect their sample, and then a smattering of other ways that people were able to access lakes. So about 50% went to their pre-assigned spot, and other people went to different places. And that, again, was okay. We told them that was okay. We just wanted to know about it. We were interested in how long it took people to sample. We had suggested via our training materials that it would take 20 to 30 minutes to do the whole process. And we found that was relatively accurate. The plurality of people said it took less than 15 minutes. About 40% said it took less than 15 minutes. Another 35 or so percent said 15 to 30. And then a smaller percentage said it took 30 to 60 minutes, so a little bit longer. But most people were able to do it in less than 30 minutes. Again, feedback on our instructions. Most people thought that the written instructions were very useful, and even more thought the videos were useful. So that's good to know. We did get some suggestions for how we could improve, including creating a separate troubleshooting video. And I think we have information on that now of what kinds of problems people had that we might want to anticipate. One of the things we learned is that, like I mentioned earlier, there was some confusion on which components of the kit would be shipped to which places and what container they went in and what shipping label and all this stuff. And so creating a video on how to properly return equipment seemed like a really great suggestion of how we could improve that in the future. And here's just a couple more points on feedback from volunteers. So again, including those troubleshooting tips. Now that we know what the troubles are, we can anticipate that for any future sampling. Sending the sampling kits sooner, that definitely resonates with me. The sampling kits went out relatively close to the time window, and I know a lot of people were moving between, lake homes or other homes, or weren't going to be at the cabin where they could receive the kits. So getting those kits together and out to volunteers sooner seems like a great idea. More tips on how to avoid contamination, potentially related to those troubleshooting tips, is another point that we took to heart. And then encouraging volunteers to work with someone else for additional assistance. Like I noted, only 42% of volunteers sampled with a partner. We will really encourage, if there's any future work in this realm, to work with someone else. It can just help for when you're trying to avoid contamination, to opening up the bags and things like that, and avoiding touching the materials when you're not supposed to. So what did we learn from all this? Number one, volunteer monitoring works. Y'all were amazing with your response rate, with your detectability, with your low contamination rates. Really highly promising results in terms of how well volunteer monitoring can work with eDNA. So kudos to all of you for that. We confirmed that spiny water flea are the most difficult to detect out of our target species using eDNA, and zebra mussels are on average the most detectable. So that could be useful when designing any future monitoring. We have ideas for how to tweak and improve our training materials. And I know a lot of you are interested in options for how this might move forward. We are exploring options and thinking about possible future directions, given the promising nature of these results. So again, my kind of motivation as a researcher from the get-go on this was the idea that there's so many lakes out there, and that eDNA might be a way to sample a large number of lakes for the presence of different AIS. Still interested in applying that more broadly, but it is pretty heavy logistical and financial and planning lift to figure out how can we get these kits out to a number of people, and most importantly, who's going to analyze the DNA once those samples are taken. So this is not something that I have answers to today, but it is something we're interested in and considering. So that's all I have. Just want to wrap up by once again saying a huge thank you to all of our volunteers. This was an amazing result, and I'm so excited at how well people responded in terms of both the results of how well things were detected, but probably more importantly, how the positive response to wanting to do this and being engaged in the process. We are interested in seeing pictures of you in the field or getting any other feedback. Here is the project email address that you can feel free to send any of those pictures to, or post it to social media. And if you're interested in more information, as Maddie said in the beginning, this webinar will be posted on the YouTube page. We have a website linked here that gives you all the information that we have, including our training materials. And we're happy to answer any other questions that might come up now or going forward. So with that, I think we have lots of time for questions. So thank you for listening and for your work on this project.
Oh my gosh, Gretchen, I love this project so much. I am thrilled that you were able to present. And we have, first question is, we have an individual who works for a small environmental nonprofit out of Southern Ontario. They were hoping to implement some reusable sampling materials in our eDNA kits that they send out to the community members. Do you have specific reusable equipment that you would recommend for this, whether that be filters, cartridges, pumps?
Yes, so the filters are not reusable. They're kind of self-preserving filter kits that we purchased, but the pumps and the water bottles and stuff like that are definitely reusable. That's what we got back. So in order to reuse them, we would have to, run them through our bleach protocol and everything like that. But that's was kind of our hope, that we could reuse the bottles and the pumps that we used, and the tubing and all that stuff. So happy to share our equipment list. I think most of it is on the website, but I'd be happy to answer any more specific questions about where we got the materials or anything like that.
Awesome. All right. So we have another question that popped in, getting into a little bit of the results. Does the probability of detection relate to the abundance of species in a given lake or the specific biology of the species, or is it a combination of both?
Yeah, great question. Yes, it's a combination of a number of things. So I used to have a schematic of this. I don't think it's in this presentation, so I'll just say it with words. To detect eDNA from a sample, you can imagine, a species has to be present in the lake. It has to be shedding DNA, so active in some way. And then those DNA molecules have to be in the water sample that is taken. So we're talking about relatively low volumes of water here. 250 milliliters. Sometimes we had 500 milliliters, but relatively small volumes of water. So a molecule of DNA from the target species has to be in that water that is then filtered and then sent to the lab to extract and amplify the DNA. So if the species isn't present at all in the lake, then it's unlikely that DNA would be there, although there are ways that it could be. But even if the species is present, you might not get it in your water sample, right? And that's, you can kind of imagine in a huge body of water that any given water sample might not have the DNA of every species that lives in there. So it is really normal. Like, I would never expect 100% detection rates for most of these species when you're taking this tiny volume of water relative to what's out there. So that's why most eDNA sampling protocols involve taking multiple samples or high volumes of water just to increase your probability of getting it if it's there. Does it relate to abundance? Yes, there's a lot of research on how well can we relate like the amount of DNA that comes out of a water sample to the abundance of the species that's there. It is related to the abundance, but it's also related to what are they doing at that time. So like rusty crayfish, for example, go through a molting period where more DNA is produced in the water. Common carp, when they're reproducing in the spring and spawning, there's a lot more DNA in the water than there is in the fall. So it is related to abundance, but not very tightly in a lot of cases. That was a lot of parts of the question, so hopefully I got all of it.
No, thank you. Okay, so kind of along that same line, if you, let's see, if you have a positive that does not repeat in additional analysis in the lake, not known to have that AIS species present, what would be the process to prove whether or not it has that AIS species?
Okay, so let me make sure I tracked that because I'm not looking at the
So
question.
yeah, it's the positive hit in an area that where a lake is unknown to have that species. How do you determine or prove whether, it was a false positive or if that has that species truly there?
Right. Yeah. So again, that wasn't the focus of this study. We weren't looking for new species. We were just assessing the probability of detecting known species. But the question is about if you did get a positive hit on a species you didn't think was there, how would you interpret it? It is impossible to prove a negative, but and these protocols are developing in kind of the AIS world of how do you treat a single eDNA hit? How many times do you need to see it? And that's going to really vary based on kind of individual or organizational kind of risk perceptions, I would say. I think given that eDNA is so highly sensitive and prone to contamination from a number of different sources, I would not treat a single one hit of eDNA as, proof that a species is there, but I would treat it as potentially a reason to go keep looking for more. But again, kind of how you interpret probability and risk is a very personal / organizational choice, and there's not any one kind of right way to interpret it.
All right, next question. Were there detection differences between nearshore collection, thinking about public access, or the open water collection from a boat?
There were not in our phase 1 study where we had like a lot of paired samples to test that, where we did nearshore and boat samples with the same person at the same day and all that stuff. So that's why we told people it doesn't matter, like your assigned site is from the shore to make it just easy and accessible to anybody, regardless of whether you own a boat or property on the lake. But if you're on a boat or you own property and you want to do it somewhere else, that's fine too. We didn't test in the volunteer samples the difference between offshore from a boat versus nearshore samples, because they weren't designed in that sort of paired way. It was just sort of ad hoc of where people decided to do it. So we didn't necessarily have, this lake for this species, we've got a boat sample and an onshore sample from the volunteer project. But from phase 1 we did, and we found no difference.
All right, we have a couple of questions actually relating to the cost of the sample kits, as well as the cost of actually processing the samples.
Okay. Is that it? What is the cost of the kits?
Yes, sorry. Yep. How much do the tests cost, as well as how much do the kits cost to put together?
Yeah, so okay, that I should have that number, and I don't off the top of my head with all of the components of the kits. The most expensive part of the kit is the self-preserving filter pack, and those are about, I might get this wrong, I would have to double check. Those are about $25 each, I think. Don't quote me on that. I could look it up and find out. But that doesn't account for the other components of the kit, like the tubing and the pump and all that. But those were relatively cheap. I don't have a number right now of exactly how much the components of the kit cost, but I could come up with it pretty quickly. And then also on the sampling side, the lab side, that's a challenging question to answer, because for this project, we used university labs who do this kind of eDNA extraction and quantification in the labs as part of their research. That's not quite the same as a lab that might be looking for contracts for hire, like a commercial lab. And most university labs are not necessarily set up to be contract for hire labs. So that's part of the challenge of thinking about if somebody wanted to implement this on a broad scale, who would do that lab analysis and how much would it cost? It wouldn't be the same people who did our analysis, because these are like university researchers. We had, grad students running samples as part of their thesis research, which is just a different ballgame than doing kind of a widespread commercial lab operation. So that's a really unsatisfying answer. But I can tell you how much the kit costs I just don't want to get it wrong. So I want to actually look at our budgets, but the lab question, I can't really answer.
Considering that, is there any chance of the U of M being able to take over this in-house, or thinking about even the Genomics Center?
I definitely cannot speak for the U of M or the Genomics Center to answer that question. I mean, is it possible? Probably. But these are the kind of larger scale logistic financial questions that need to be answered that I don't have the answers to.
All right. Okay, we're going to kind of jump gears. Does your research team have a plan to do more focused monitoring on lakes with brand new AIS detection, newly named zebra mussel infested lakes, to test the sensitivity of this process on a less established population?
That's a good question. We don't currently have a specific plan of where we want to go next, but we are considering the different ways right now, kind of what are the big unanswered questions, and where are we, like me, my lab, or collaborators, or MASERC or the University of Minnesota, who's well positioned to answer those unanswered questions. And that's kind of a work in progress still as we take in these results and figure out what to do next.
Will this sort of testing work for starry stonewort?
Ooh, starry stonewort. That's a good question. I would have to double check the literature. So in general, aquatic plants, in terms of their eDNA, like the primers that you need to be able to identify their DNA in the lab, the plant DNA has lagged a little bit behind some of these others, but from what I understand, there is increasing use of eDNA for aquatic plant and macroalgae detection. I think starry stonewort has been the target of eDNA sampling, but not in my lab, so I don't know for sure.
Just a question about the procedure. They mentioned that they might have missed this earlier, but just to reiterate, were all 4 species monitored from the same filter?
Yes, well, potentially. So all 4 species, if all 4 species were known to be in the lake, then yes. But yes, you can use the same filter for multiple species.
Another method question. So you were talking earlier about the field blanks. Would it be feasible to test the field blanks from all of the volunteers rather than that subsample?
So we only gave field blanks to 10% of the volunteers, because it was an additional cost of shipping, including a large jug of water with it, and it's additional sampling for the volunteers. So we just randomly selected a subset of volunteers who did the field blanks. So not everybody did them, but we tested all the ones that we did.
Did you screen the sample with specific species probes and QRPC or metabar coding?
Yes. Yep. So this question is about, just to explain to people who might not know, qPCR can be done for species specific probes, and metabar coding refers to eDNA approaches where you can look for a whole bunch of different species kind of in one sample, even ones that you haven't that you don't know are there, or for example. So this was single species qPCR, not metabar coding. So we had these 4 target species and we tested each sample for those single species targets. We did that because in general, the single species qPCR can be more sensitive to low numbers of DNA, but there are constant improvements in the genomics world and in metabar coding and all kinds of other technologies that might make that also a feasible option, but that's not what we did. We did single species qPCR.
Awesome. Well, thank you so much for everyone's time, for especially Gretchen for giving this presentation.